Sleep takes up about 1/3 of our lives, but we still don't fully understand why we sleep, how sleep is regulated, or why sleep disorders happen. In the last decade, genetics has completely transformed how we study sleep and circadian rhythms. Today, we're exploring how your genes shape sleep timing, sleep quality, circadian rhythms, and major sleep disorders like narcolepsy, restless leg syndrome, and insomnia. Let's dive into the genetics of sleep and circadian rhythms. Sleep is a reversible state where cognition, motor activity, metabolism, and environmental responsiveness are reduced. It's universal across the animal kingdom, which tells us that sleep is ancient and essential. In humans, sleep is divided into two: NREM sleep, which supports physical restoration, immune function, and memory consolidation; REM sleep, which drives emotional processing, creativity, and neural rewiring. We identify these states using EEG, which records brain waves, and EMG, which measures muscle tone. But why do we sleep when we do? And what determines whether someone is a morning person or a night owl? To explain that, we need to look at two major biological systems that regulate sleep. Since 1982, sleep has been understood through the two-process model. 1. Process C, the circadian system. This determines when you feel sleepy or awake. 2. Process S, sleep homeostasis. This controls how much sleep you need, based on how long you've been awake. A healthy sleep cycle depends on both systems working together. If they fall out of sync due to genetics, lifestyle, or environment, sleep disorders can emerge. Circadian rhythms are generated by a molecular clock inside almost every cell of your body. At the center of the system is the suprachiasmatic nucleus, or SCN, the master pacemaker in the hypothalamus. Most cells in your body have their own clocks, but the SCN synchronizes them to the 24-hour light-dark cycle. Inside each cell, the circadian clock is built from translation feedback loops. The transcription factors clock and BMAL1 bind together and activate key expression genes, including PER1-3 and CRY1-2. After PER and CRY proteins are made, they pair with the kinases CK1 delta/epsilon, move back into the nucleus, and turn off clock BMAL1 activity. After they degrade, the cycle restarts. Roughly every 24 hours, the circadian system adapts to environmental signals called Zeitgebers, meaning time givers. The strongest Zeitgeber is light. Light is detected by photosensitive retinal ganglion cells. They send signals to SCN neurons, activate CREB-mediated PER transcription, and this resets the clock to the day-night cycle. Other Zeitgebers include feeding time, glucocorticoids, body temperature, metabolic state, and sleep-wake history. Peripheral clocks respond to these cues, but exactly how many of them interact with the molecular clock remains unclear. Your chronotype, whether you're a morning person or a night owl, is shaped by both environment and genetics. Morning types prefer early sleep and wake times. Extreme cases may show advanced sleep phase. Evening types naturally stay up and wake later. Extreme cases may show delayed sleep phase. Twin studies show chronotype is 12 to 50% heritable, meaning your genes play a major role in how your internal clock runs. Here are some fascinating human mutations. CRY1 mutation, delayed sleep phase disorder. People naturally fall asleep 2 to 4 hours later than typical. PER3 gene variants, extreme morningness. A long repeat in PER3 pushes the clock earlier. December 2nd mutation, natural short sleeper phenotype. People with this rare variant sleep only 4 to 6 hours but feel fully rested. These examples show how small genetic changes can dramatically alter sleep timing and need. Chronotype is shaped by age, sex, environment, light exposure, and genetics. The homeostatic system regulates the amount and quality of sleep. In adults, the typical sleep duration ranges from 7 to 9 hours, following a normal distribution, but genetics strongly shapes these traits. Heritability estimates include sleep duration 30 to 65%, sleep latency 33 to 83%, sleep EEG architecture 50 to 96%, daytime sleepiness 17 to 48%, and daytime napping 65%. So while lifestyle matters, your baseline sleep tendency is strongly genetic. Because circadian rhythms and sleep traits are moderately heritable, human genetics gives us powerful tools to uncover the molecular basis of sleep and circadian timing. This knowledge has major practical implications, including better classification and diagnosis of sleep disorders, improved understanding of neuropsychiatric and metabolic diseases, more personalized approaches to treatment and targeted interventions for circadian misalignment. Large-scale genome-wide association studies (GWAS) have begun uncovering the genetic basis of many common sleep disorders. Let's look at 3 major conditions. Narcolepsy is a rare, usually adolescent-onset disorder that affects about 1 in 2,000 people. It involves excessive daytime sleepiness, REM sleep dysregulation, vivid hallucinations, and in narcolepsy type 1 cataplexy, it's a sudden muscle collapse triggered by emotion. Genetically, narcolepsy is fascinating. First-degree relatives have a 20 to 40 times higher risk, and GWAS and exome studies have identified 8 genomic regions, 3 specific genes, and 9 HLA types. The findings strongly support autoimmune and inflammatory mechanisms, especially for narcolepsy type 1. Restless leg syndrome affects 5 to 15% of adults. It is marked by an uncomfortable urge to move the legs, symptoms that worsen at night, and relief through movement. Genetics is a major factor. 60% of cases report an affected family member. Early onset RLS typically appears before age 45. GWAS have identified 19 risk loci, plus 6 more from transcriptome-wide studies. These explain about 12% of the genetic variants, showing the disorder is strongly polygenic. Insomnia is a sleep-wake disorder characterized by chronic difficulty initiating or maintaining sleep, or by non-restorative sleep, accompanied by daytime symptoms such as fatigue or impaired functioning. Insomnia disorder is defined as symptoms occurring at least 3 nights per week for at least 3 months, affects approximately 10% of the population, while occasional insomnia symptoms are reported by roughly 30%. Family and twin studies estimate heritability between 22 to 59% in adults and 14 to 71% in children, indicating substantial genetic contributions. Large GWAS have identified many associated loci, but the overall SNP-based heritability is about 8%, suggesting that insomnia is highly polygenic, involving many small effect variants, and few specific causal genes have been confirmed so far. Misalignment between internal clocks and external schedules has significant health effects. Chronic circadian disruption is linked to metabolic syndrome, type 2 diabetes, cardiovascular disease, depression and bipolar disorder, impaired immune function, and cancer risk. This happens because 2 to 30% of each tissue's transcriptome follows a circadian rhythm, meaning timing affects nearly every biological process. One of the most common circadian problems today is social jet lag, which is the difference between your biological clock and your social obligations. Example: natural sleep 12 a.m. to 8 a.m. Work schedule 7 a.m. to 3 p.m. This mismatch leads to poor sleep quality, metabolic disturbances, mood problems, and lower cognitive performance. Genetics determines your chronotype, but society often forces a different schedule. Sleep and circadian rhythm biology are entering a transformative era. Genome-wide studies find associations, not direct causes. The next challenge is functional biology. Researchers now aim to test how these variants affect neurons, circuits, and molecular pathways, study cell type-specific and developmental stage-specific effects, integrate multi-omic data, genomics, epigenomics, proteomics, metabolomics, and translate discoveries into clinical tools and therapies. The ultimate goal is to understand how sleep works at the molecular level and use that knowledge to improve human health. Sleep and circadian rhythms may soon join cardiovascular disease and cancer as areas where genetics revolutionizes diagnosis, risk prediction, and treatment. Here are a few simple science-backed ways to support your circadian clock. Morning light exposure stabilizes your clock. Consistent sleep and wake times strengthen sleep homeostasis. Avoid late-night eating, it confuses peripheral clocks. Reduce screens at night, blue light delays melatonin. Honor your chronotype when possible. Protect sleep as a biological necessity, not a luxury. Your genetics set the foundation for how you sleep, but your daily habits are the tools you can control. If you enjoyed this deep dive into the genetics of sleep and circadian rhythms, hit like, subscribe, and let me know in the comments: are you a morning person or a night owl, and do you think it's genetic? Thanks for watching.